as a lecturer in the Provost Graduate Student lecture series this fall. We have a very large and highly competitive group of applicants, and the selection committee has a difficult task of culling them down to just a few. We think this series will be really interesting for everybody because it's really very broad in its outlook. We're starting with neuroscience, but we've got somebody from physics, somebody from sociology, and somebody from fine arts. So it's a real expression of the diversity of achievement of graduate students here. And we'll all get notices, hopefully, for each of the upcoming lectures. So traditionally, we let the advisor do the praising introduction, so I'm going to let Mallory take over.
Well, it is my pleasure to introduce our speaker today, Mallory Locklear. We were discussing on the way over here what year she was in her graduate program, and we decided what we were going to go with is very sad to me, she's a last year graduate student, which is devastating for me. In addition to the fine work that you're going to see here, which is really just a fraction of what she's done and is doing in our lab, Mallory is extremely active in the university and graduate school. She is the secretary of the GSO. She is a member of the Stony Brook Council. She's certainly a valued member of our graduate program and one of our ACE recruiters, and always there to lend a hand in the department. So she's been not only a wonderful researcher to have. I hate to talk about her in the past tense. I'll start to get misty here and panicky, actually. But she's just been a wonderful citizen in our program. What she's going to talk to you about today is, again, just a fraction of the work that she's done. And I think, and I will tell you, Mallory, this is not published, although it will be shortly. And I think when it gets out there, so you are the first to hear this story, and I think it's pretty exciting, and some of the results are fairly jaw-dropping, I think, and we hope that you agree. And we think that the world at large is going to be actually quite interested in what Mallory has to say, which is that males are different from females. Who knew? Mallory.
Thank you, Mary. So yes, that is what I wanted to tell you today, that men are different from women, which seems like obvious, right? Seems a very obvious thing to say, but we in the neuroscience community have been remiss, and we've been ignoring that fact for quite some time, actually. So most of the experiments we do in both humans and animals focuses on one sex or the other, usually. And usually that sex is males. And it turns out, again, males are not the same as females, and we have been doing ourselves a disservice. So what we did in the lab, so we started looking at both males and females to see, are there differences? And turns out there are. So I'm going to talk to you about this in respect to disease, dopamine, and my favorite part of the brain, the prefrontal cortex. So I would like to start there, the prefrontal cortex. And this is the prefrontal cortex. It's part of the forebrain. And as you can see, dopamine, the neurotransmitter I just mentioned, is produced here in a different part of the brain. And the cells that produce this transmitter, dopamine, project up the prefrontal cortex and drive it and innervate it. And the prefrontal cortex, to be healthy and functional, really requires the right amount of dopamine to come from this area. That's actually only half of the story. The other half of the story is that the cells that are receiving, or the area that is receiving this dopamine innervation, the prefrontal cortex, those cells are projecting right back down to the area that is producing this dopamine. And it's the communication between these two regions of the brain, the prefrontal cortex and this region called the ventral tegmental area, that is really key to the prefrontal cortex being healthy and functional. And when dopamine goes awry or something goes awry in the prefrontal cortex and communication breaks down, it results in pretty hefty problems. So the executive functions of the prefrontal cortex. So what is the prefrontal cortex doing that is so very important? Well, here is just a small list of those things. Planning, problem solving, attention, behavioral flexibility, working memory. These are all really important to being a productive citizen, being a productive human. If these things are going wrong, you're probably experiencing some pretty poor quality of life. So I want to give you an example of differences between the way men and women really express these executive functions. And this is a very scientific thing. Mission, go to Gap, buy a pair of pants. So this was given to me by one of my committee members after I showed him some videos I'm going to show you. And as you can see, the male here goes directly to the Gap, 6 minutes, $33. Whereas the woman goes all the way around and spends much more time and much more money. So this is a sort of comical difference between men and women. But in fact, there are actually some similarities that we can actually measure in animals. So the way I do my work is I look at animal models, and you can actually go through and look at these executive functions of the prefrontal cortex and observe them and measure them in an animal model. And we do this in the Barnes maze. So the Barnes maze, I'm going to give you a sort of brief introduction to this task. What you're seeing here is a top-down view of the Barnes maze. It's an elevated table, about 3 feet across, and as you can see, there are holes all throughout the edge. And you can barely see, but I'll highlight it later. There's one hole that contains a little escape box. And if you put a rat on this table, its natural inclination is to get away from the open space and the lights and to go to this dark, confined little area. And so when you put the rat there, that's what its goal is to do, is to go find that area. And throughout multiple trials, what you would hope to see is that the rat can find this area quicker and quicker and more efficiently. So the first time you place a rat on this table, excuse me, it will probably take some time to locate the hole because it has 12 to choose from. And it's going to go through and investigate, and eventually we'll find this hole. Now around the room, you should know there are very large shapes and different colors, and they're used to help direct the rat to where they need to go. So they can orient themselves in space based on these different cues that we've placed on the wall. So at the time this rat has been on this maze multiple times, hopefully this rat is using some sort of strategy to get to that hole much more quickly than it did at the beginning. And what I'm going to show you is that the female and the male does this through very different means. So they're both utilizing these executive functions of the prefrontal cortex, which we are testing on this maze. They're using them well, they're getting to the goal, they're just doing so in entirely different manners. And so I'm going to show you our version, our rat version, of the mission, go to Gap, buy a pair of pants. So the gaps are the goal. Okay. You can see she goes to the wrong hole. He goes to the same wrong hole. Now watch how they solve this problem differently. He is insisting this is the right hole. He's looking around, using his spatial cues, knows he's right in the right area. And there he goes. Now he finally finds it. Whereas the female, as you have seen, has gone around the entire maze and gets there in almost the same amount of time as the male. So you can argue who does this better. But the point is, they get there at the same amount of time. They're both using their prefrontal cortex, and they're really showing these executive functions. They're working for these animals. They're just working in completely different ways. And this is what we're interested in. So let's talk about what is driving these differences in the executive functions of the prefrontal cortex. Here's a more comprehensive list of some other things, other executive functions, social cues, learning. These are all really important, and they're all really dependent on the dopamine coming from this area of the brain. The dopamine that is leaving this area of the brain, the cells that are in this area of the brain and projecting to the prefrontal cortex, releasing dopamine, are responsible for the healthy function of these functions. If this goes wrong, so do these. And this is not a case of where more is better, because actually dopamine needs to be in a very small window in order to be functional. It's in this range that the prefrontal cortex's function is at its optimum amount, and when you fall too low or fall too high, the prefrontal cortex and the executive functions it mediates also fall down to reduced levels. When dopamine falls into these levels and when executive functions go wrong, you can see these in a variety of diseases, and I've only listed about 4 here. Anxiety, depression, schizophrenia, Parkinson's disease. These are all really devastating problems to people who are suffering from them. And they all involve dopamine being outside of this narrow window. What I would also like to point your attention to is the difference between men and women. So in all of these, there's a difference. In anxiety and depression, females are much more affected than our males. And in schizophrenia and Parkinson's disease, males are much more affected than females. And I would like to also point out that the ones that affect the females more, anxiety, depression, usually involve dopamine being too high, whereas males are usually experiencing diseases where dopamine is too low. So that begs the question, what makes men and women more susceptible to hypo, too low, or hyper, too high dopaminergia? And our first thought was, well, maybe males and females start off with different levels of dopamine, period. And so at resting rates, when everything is going fine, the prefrontal cortex is functioning well, maybe the men and women have slightly different levels of dopamine, and that they're both within this optimal range that the prefrontal cortex needs and likes. But maybe women are just poised to the edge, the threshold of being too high, whereas men are more on the threshold of being too low. That's the first thing we looked at. It's just at basal rates, is there a difference? I'm going to tell you the answer, and I'm going to tell you how we got there, but the answer is no. In fact, that is not the case. So let's look at how we did that. So the prefrontal cortex has a variety of neurotransmitters, but the one we're focused on right now is dopamine. And the way we go about measuring this is through this setup. This is called microdialysis. So we have a rat, and what we do is we put a tiny little tube directly into the prefrontal cortex of the brain. And in that tube, we can put a probe, which you see here. And on the end of this probe is a little membrane, and it allows us to deliver fluid, but also sample out fluid, and we can measure these neurotransmitters directly. So the fluid we'll be putting in are artificial cerebrospinal fluid, which just mimics the natural fluid of the rat's brain. Or we can put in a drug. When we put in the drug, we can see what happens to dopamine, see if we can change it. Now, once we have put in these fluids, we can measure the neurotransmitters in vivo by sampling them back out. When you sample out the fluid, you can measure the neurotransmitters, and you can get the concentration of dopamine. And so we do this in live rats. They're usually asleep in order to keep movements from being an issue. But I also like to point out that they are healthy, happy rats. I had to wait this one up for the picture, so she wasn't too happy with me, but they are functional living animals that we were measuring from. And again, what we found was that dopamine at basal rates, when nothing else is going on, when these rats are healthy and functioning rats, there isn't any difference between dopamine in males and females. You see a small difference, but it's not significant. So in fact, it is not that the females are starting off too high and the males starting off too low. They are actually right on top of each other. So if it's not dopamine starting off too high, what is it? Our thoughts were, well, maybe the mechanisms involved to keep dopamine in this narrow window, maybe those are different between males and females. So again, here is the circuit we were talking about. You have the dopamine cells from the ventral tegmental area projecting up to the prefrontal cortex and releasing their dopamine. The cells in the prefrontal cortex are projecting right back down to the dopamine area, and this communication is what is key to prefrontal cortex function. And what I'm going to tell you is that yes, there are differences in the way this communication works between males and females, and that is what's causing these differences. And in fact, it's actually this part of the circuit where this is occurring. The projections from prefrontal cortex to the ventral tegmental area. And so let's talk about the circuit in a little more detail. So this is just another representation of what you see here. These are the cells in the prefrontal cortex that are projecting down to the ventral tegmental area and are driving these dopaminergic cells in this area. So these dopamine cells are then projecting right back up to the prefrontal cortex, and they're driving each other. Now, to keep these things from going too crazy, we have neurons within the prefrontal cortex that are releasing a neurotransmitter called Gaba. This is an inhibitory neurotransmitter, and this is sort of acting like the brakes of the system. It's keeping everything in check. It's making sure things don't go too crazy. What you'll also see are what I labeled NMDA receptors and AMPA receptors. So these are two different types of receptors from the same family. They both bind to the neurotransmitter glutamate, and they both send go signals to the cell that they are attached to. So for instance, this AMPA receptor is here on the pyramidal cells, the prefrontal cortex cells that are projecting to the dopamine cells. And if glutamate binds to these receptors, it tells this cell to go, which will then tell these cells to go, which then releases dopamine into the prefrontal cortex. Now the NMDA receptors are giving the same sort of signal. They're also saying go, but here they're telling an inhibitory cell to go. So when you bind glutamate to these receptors, you tell this cell to go, you pump the brakes on the system, it calms everything down, and dopamine should go down. What I'm going to show you is that this is not the same for males and females. So what I've showed you is the male version. So what's going on in the females? Well, up until now, most people did not know. Again, everything was done in males and just presumed to apply to females. It Turns out that is not the case. So I'm going to show you the differences, and then I'm going to show you how we got there. So do you see the main differences here? We have the same players, we have the same cells, but now these important NMDA receptors that are a break in this system have now moved over here and are now giving a go signal to this cell. So now instead of telling the brakes to go, it's sort of telling the gas pedal to go. And so what you see is that the male, it's telling things to stop, and the female, it's telling things to go. So let's see how we got there. So we turn back to microdialysis. So this is one of our microdialysis experiments. The way this is reported is here's the time of the experiment in minutes. We also have dopamine as a percent of baseline. So what that means is I took basal levels of dopamine from these animals, made sure they were steady, they weren't changing on their own, and then we applied a drug to see what would happen. And this drug is the NMDA, so we're targeting those red receptors from that previous slide. And this is showing how long the drug was on, and this is showing how much it changes from baseline. So in the male, where those receptors are the stop, the brakes of the system, you put in NMDA and dopamine goes down just like one would expect. However, in a female, it does the exact opposite and increases dopamine by about 40%. So this is really showing us that those receptors are giving two completely different signals to this system. One is encouraging dopamine to go, and one is keeping dopamine from going. So let's look at this in our study. So again, this is what I just showed you. And this is really highlighting the differences we're seeing here. So this is really showing us that NMDA is driving the stop signal here, but is driving a go signal here. And so what you see now is that this cell has way more go signal than this one, right? Has only AMPA over here telling it to fire, whereas in the females we have both NMDA and AMPA telling it to fire. So you would expect this cell to be going crazy. It should be firing way more than in the male because it's getting so much more input telling it to go. And if that were the case, it would now be telling those dopamine cells to go crazy as well, and these dopamine cells should be firing way more than in a male. And this would result in far more dopamine being in the prefrontal cortex of a female versus a male. But if you remember, that's the first thing we thought would be the difference. And I already showed you that that is not the difference. Dopamine is actually the same between males and females, so what else could be going on to keep this in check? Well, let's look at this third player that we've been ignoring a little bit over here. The cell that is releasing GABA, the brakes of the system. Our thought was, in order to keep these cells, which are just ready to go crazy, to keep those in check, maybe the brakes of the system are much stronger than in the male. And so when you increase the brakes on the system, you're really able to control everything back down to normal levels. So how do we test that? Well, the way we tested it was we completely got rid of the GABA signal. We just blocked it. So same with microdialysis, we put a drug in that just takes away that GABA signal and it's not there anymore. So we've taken the brakes off of the system in both the male and the female. And if what we think is correct, when we take the brakes off, what should happen is dopamine should increase in both, but it should increase much more in a female. Because again, the system is just ready to go. This is the only thing holding back this cell that just has so much go signal. And this is what we found. So again, microdialysis, you have time in minutes and percent of baseline here. And when we block the GABA system, we take the brakes off, you see that in a male, dopamine increases by about 2-fold. And it stays up a fair amount for the course of the experiment. And in a female, it goes up way, way more. It's almost 600 times the normal dopamine level when you take the brakes off the system. And this is really giving evidence, it's supporting our idea that it's the extra brakes in the female that is really keeping everything in check. So this is now much bigger compared to this in order to keep this system in check. So what we wanted to do to make sure that is what we were actually seeing is do electrophysiology, so we can actually go in and record these cells firing. We want to see that if this is the case, that there is the same amount of dopamine between males and females, then theoretically, these cells should be firing at the same rate. Because if they were firing differently, that would result in different levels of dopamine being in the system. And this is how we do that. So you have a rat, and you can lower an electrode into the brain area of interest. We can do this in both the prefrontal cortex and in the ventral tegmental area, and get both of those types of cells. All right, our signal is then amplified, so we can actually see it. And then we can read it out on a computer, and you can also listen to it. And this is what the output is. So this is an example of a little bit of a dopamine cell. And if you zoom in on this right here, this is what the action potential looks like. And again, if dopamine levels are the same, the firing rates in both the prefrontal cortex and the VCA should be the same. So here is a sample of our ventral tegmental cells. So these are the dopamine cells, those cells that are projecting from ventral tegmental area up to prefrontal cortex. And we can tell they're dopamine based on the shape of this action potential. And as you can see, they are remarkably similar between males and females. I promise these are actually different records. This is not me just copying and pasting. And as you can see, the overall activity, so this is frequency of firing, how often these cells are firing their action potentials, are remarkably similar between a male and a female. Now, the ventral tegmental area does not only have dopamine cells, it also has other sorts of cells. And one of the other ones we can actually tell what it is are Gaba cells. And just to show you that things are really just working the same in males and females, I'm showing you some of this data as well. So again, we can tell this is a Gaba cell based on this action potential. They fire much faster than dopamine cells. But again, you can see there really isn't a difference between the males and the females. So that region of the brain that is producing that dopamine, that is driving the prefrontal cortex, everything in there is behaving just the same between males and females. So let's check out the prefrontal cortex. This is an example of the pyramidal cells we're measuring. So these are the cells that are projecting from prefrontal cortex down to ventral tegmental area and really driving those dopamine cells. And again, you can see between males and females, they're pretty the same. And again, if you look at their overall activity, the frequency of these cells between males and females, very similar. So let's go back to our original question. So I've showed you a lot of things that are the same. You know, at the beginning of the talk, I told you things were wildly different, right? So what makes men and women more susceptible to these diseases? Why are women far more susceptible to diseases where dopamine is too high? And why are men far more susceptible to diseases where dopamine is too low? As I again told you, they're all at the same level when things are working perfectly. Well, the answer sort of lies with these differences I did point out. So, again, just to go over the circuit, we have pyramidal cells from prefrontal cortex projecting to and then driving dopaminergic cells in the ventral tegmental area. And these cells are projecting right back up to the prefrontal cortex and driving the system. It is the prefrontal cortex, the health of the prefrontal cortex that relies on this system working properly. And in the male, we have a fair amount of brakes on the system, and in female, we have a lot of brakes on the system, really holding back all of this. So that's what we think the difference is that the female is really functioning because this cell has a chokehold on a lot of dopamine. There is potential for this cell to go way too crazy, and a potential for these dopamine cells to then release way too much dopamine in the prefrontal cortex. So if anything goes wrong in this system, which is so perfectly balanced in a normal animal and in a normal human, if anything goes wrong, it is most likely going to lead to way more dopamine being released in the prefrontal cortex, because that is what this system is set up to do. It is this that is keeping that from happening. Whereas in a male, you have a fair amount of signal coming through with a fair amount of dopamine coming up. There's really not much being held back. It's sort of already at its maximum. And so if something goes wrong, the default is to fall too low. And that is where we think the difference is between males and females, and why one is more prone to one disease and why one is more prone to the other. So what's the answer? Why is this happening? What is causing this? And how can we fix it? Well, we think, our lab thinks that it has to do with hormones. So if you've noticed, there's a little black dot in this cell. And what this represents is the massive amount of androgen receptors that are on this cell. So the receptors that are binding testosterone are primarily on this cell in the circuit and nowhere else. And in a male, you will obviously have far more androgen than in a female. So if we show that here. And what we believe, based on some other experiments we've done, is that androgen is keeping these receptors out of the cell. That is why you see a signal only from AMPA receptors here. Whereas in a female, you have much less androgen, and it's allowing these NMDA receptors to be expressed in this cell, which leads to the difference you see here and here. Now, estrogen also plays a role. So estrogen is actually located here on the brakes of the system. Many of these cells in the prefrontal cortex express a lot of estrogen receptor. And so in a male, or in a female, you have far more, versus a male that has far less, and that might be leading to differences that you see here, and that this cell is firing a fair amount, and this one is really pumping the brakes of the system. It's very related to hormones, and we think that's also the key in how this can be fixed. It's a more indirect way to solve problems when things go wrong than just by directly changing neurotransmitters, which can be a little messy. But if we go in and we manipulate the different hormone levels that men and women already have, maybe that's an answer to fixing these problems that have been so hard to fix for so long, like schizophrenia. They're notoriously bad for fixing the symptoms of schizophrenia. Medicines just really aren't working. So maybe if we go through, rather than directly trying to change the neurotransmitters in the system, let's mess with the hormones. Maybe that'll make the difference. And with that, I'd like to thank my lab members. So Terry was the grad student who came before me, who taught me everything about microdialysis. Asa Dayang, Alice, the second year, who did a lot of the microdialysis experiments. Amanda and Bridget did a lot of the histology to make sure that those probes are in the right spots. Our collaborators who've been really helping us with the electrophysiology, which is pretty new for our lab. Bill and Patricio at Maryland and Holly Moore at Columbia. And of course, my community members, Craig, Bill, Giancarlo, and Mary. Thank you.